All right, so now we're going to talk about active transport. So active transport, remember, active requires energy, passive does not. So when you're passively laying on the couch watching TV, you're not using too much energy. But when you are active and you're going for a run or playing football, soccer, what's happening? You are using energy. So active, the action is happening. So this is movement of molecules from a low concentration to a high concentration. What's happening? Inside of the cell, there could be a high concentration of sugar molecules. If those sugar molecules are in a high concentration, and outside of the cell, we have a low concentration of sugar molecules, those sugar molecules that might move in to the cell from the outside of the cell are moving from a low concentration outside of the cell to a high concentration inside of the cell. So we are using, or our cells are using, active transport. Now, another way you can think of this, active transport, we're walking up a hill. We're going from low ground to high ground. So, low concentration to high concentration. When we are moving from the top of the mountain down the mountain, so we could think we're walking down or rolling a ball even, it's passive. We're going from high concentration or high ground to low concentration or low ground. So how is active transport like going up a hill? So molecules are going from low. That means there's a low concentration of these molecules to a higher concentration of these molecules. So we're adding more molecules to an already high concentration. Remember, concentration deals directly with solutes and how they're dissolved in a solvent. This process needs energy. It requires ATP. And how is this performed? Through cellular respiration. So the energy, ATP, comes from cellular respiration. And the organelle that's responsible for cellular respiration is called the mitochondria. Now we'll go over cellular respiration later on, probably our next unit, and we're also going to learn about how ATP is created within the mitochondria. Now, active transport most of the time needs to use molecules, and it's called a protein pump. A protein pump can move molecules across the membrane. What happens? They're going to move these molecules that are in a low concentration to a high concentration, and they're going to move the molecules through something called a protein pump. So, like turnstiles at a stadium, it pumps admits certain molecules into the cell. Okay? Remember, active transport goes both ways, into the cell and out of the cell. So if a molecule is moving from low concentration to high concentration, whether that be into the cell or out of the cell, active transport must be used. So protein pumps, for example, will be used. The energy that's needed to operate the pump is used to change the shape of the protein pump during operation. So that means allowing for the molecule to move through, almost like a Pac-Man, and then it turns and releases the molecule. It also moves molecules through the pump from a low concentration to a high concentration. So in our little GIF below, we have an example of active transport. Some particles enter the cell without actually crossing the membrane. How does the cell do that? Through something called endocytosis, where the membrane, so remember that phospholipid bilayer, the membrane, will actually form a vesicle and engulf the molecules that it is trying to bring into the cell. So this endocytosis, we think enter the cell. So en, just like enter, endocytosis, enter. And this material that's being engulfed to the cell can either be a solid or a liquid. So this is called a membrane sac, and we call it a vesicle. So the membrane pinches off, and forms around the material to bring the molecules into the cell. So here's a really good example. We have or picture, I should say. We have the cellular membrane, and it's extending, so it's forming a sac. If you see in our second picture, the sac is formed, the cell membrane is formed around it, and the cell membrane will reattach to each other, forming those bonds, and the vesicle will be released into the cell. Remember, endocytosis is to move into the cell. There are two types of endocytosis. One is called phagocytosis, so that's where the cell is eating, and that's going to be large food particles coming into the cell. Penocytosis is the cell is drinking, so that's tiny droplets of liquid coming into the cell. So phagocytosis, you can think food. Penocytosis is water or liquid droplets. Okay, now we can look at the different types of engulfing that happens inside of our cell. We have in the first picture on the left a white blood cell attacking a parasite. So what are they doing? They're forming vesicles around the parasite. We have an amoeba eating on the top right-hand side, and you see the amoeba. It's actually forming around the food particle through phagocytosis and bringing that food material into the cell. Exocytosis, so we had endocytosis entering the cell, exocytosis exiting the cell. So this is going to be the opposite of endocytosis. Exocytosis, now the cell is taking these vesicles, so the vesicles that are within the cell, and they are moving to the cell membrane, and the material is being released out of the cell. So exit, exo. So during exocytosis, a vesicle that contains substances will fuse, that means attach, to the cell membrane, expelling its contents, so letting go the contents that's within the vesicle to the outside environment, so releasing the material outside. Now, this could be an example of how the cell gets rid of material that could not be digested. So exocytosis is a form of egestion, exiting, leaving. Another example of exocytosis, when proteins are made by a cell, are released or exported to be used elsewhere. These proteins are referred to as secretions. Examples of secretions could be digestive enzymes, mucus, saliva, and poisons. So remember, when your body, let's say you get bit by a spider, your body wants to get rid of the toxins that the spider released under your skin. So your cells are going to engulf their toxin and try to release it outside of your body or outside of all of the cells. And that's why it becomes itchy and inflamed, so that your body can release the toxin. Now, what organelles are responsible for protein synthesis? Let's remember that ribosomes are very important for protein synthesis. And we're going to look at a diagram in just a moment. Step 1, the protein to be exported is synthesized. So step 1, proteins made at the ribosome, so that means synthesized, they're made, and they're going to attach to the endoplasmic reticulum. In step 2, a part of the endoplasmic reticulum breaks away, forming a vesicle. So step 1, the protein is made. Step 2, the vesicle forms around the protein. Step 3, the vesicle moves toward the Golgi body, where it will be packaged and exported. So here's our Golgi body. Step 3, we have the vesicle attaching to the Golgi body, and we know the proteins are going to be packaged properly and then exported. In step 4, a small part of the Golgi body breaks away, forming another vesicle that's going to move towards the membrane. This vesicle will be responsible for fusing with the cell membrane, and the protein is released outside of the cell. So here, step 4, we have the new vesicles forming, and the vesicle will then fuse to the cell membrane and release the contents that was inside of the vesicle out of the cell.